The overall goal of this approach is to model liver colonization by metastatic tumor clones generated by colorectal cancers, and to use this model to improve diagnostics and therapy of liver metastases.
Clinical observations indicate heterogeneity of metastatic disease. Our model provides an ability to capture this heterogeneity, to detect genes which are associated with oligometastatic and polymetastatic disease, and to use this knowledge for improvement of early detection and treatment of metastasis.
The main advantage of our technique is that we use double-labeled monoclonal cell lines derived from patients with colorectal cancer and can observe development of liver metastases both in vivo and ex vivo.
The implications of this technique extend toward therapy of metastatic disease. It gives us the ability to test any potential drug with quantitative estimation of numbers of metastatic lesions and growth kinetics of each lesion.
After generating luciferase TD tomato-labeled HCT116 cells and preparing media, reagents, and instruments according to the text protocol, plate the transfected cells at the following densities per well in triplicate in 96 well plates. Following the incubation of the cells for 5 hours, quantify the TD tomato fluorescent intensities using an IVIS system by starting the image software on the desktop. Click the Initialize button to initialize the imaging time, medium binning, 2 for f-stop, 535 for the excitation filter, and 580 for the emission filter. Place the 90